Okay, so where we left off last, we've been talking about taking a prokaryotic gene from one species, and then we're going to insert that into another prokaryotic species. And this is something that prokaryotes do all the time. They can, they have these little, let me draw a little. There's a prokaryotic cell, and there's another prokaryotic cell. They have, these little pilices that enable them to exchange plasmid from one cell to another and amongst each other. And that's how we can get things like the infamous R plasmid that makes some bacteria resistant to antibiotics. And what enables this to happen, though, this uptake of small amounts, and I should probably clarify that it is small amounts of DNA. Plasmids are actually just 5% the size of a bacterial chromosome. But anyways, what enables it to do this is calcium ion-driven pumps, and if it's a pump, we know it's ATP-powered. This is something that they do in nature, so we can just apply them in an environment that gives them that, or we can do something called electroporation, and as the name implies, it's, I have right in here just shocking something that when you shock it, that causes holes to form, pores that enable them to uptake the plasmids that we were talking about earlier. And that's an artificial. That's something that we've done through tinkering with them. So when we do that, when we do this, when we're trying to do this, whenever we apply the calcium pumps or whatever, well, there's 4 possible outcomes. We could have naive or virgin bacteria, and what that is bacteria that have no plasmids. No plasmids. No plasmids. Bacteria that have the foreign with no foreign, DNA, the original plasmid. And what happened was basically whenever we apply these restriction endonucleases, it's going to come in and it makes a little cut there and a cut there, and what happens is it's going to separate out, that enables it to insert foreign DNA into this sequence. Well, we broke these phosphodiester bonds, and as a result of the breaking of this, these hydrogen bonds is kind of broken, split off into opposite directions. However, what happens when this is occurring, these hydrogen bonds or single-stranded DNA is unstable, so it's going to want to do, so what does it want to do? It wants to get stable, and that's something that, I think it was Einstein who just really pointed out the fucking obvious by saying that if you're at a high energy, an unstable state, what you want to do is you want to get back down here into a stable state. And so that's what they're doing. It's unstable, it seals back together and gets stable. And so what happens is the plasmid has just reformed, and it's back to its original form. There's no changes have occurred in it. And this is obviously not what we're looking for. Also, you can have bacteria where we open up with these restriction endonucleases and they get the wrong gene. They get the wrong DNA fragments. Because whenever we're applying restriction endonuclease to find the gene of interest, that's going to cut up many fragments of DNAs. And in the event of our book talks about hummingbird DNA, because we're trying to find out the beta globin gene for them because they have an incredibly awesome metabolism, but sometimes you can end up getting the entire genome of something, so it's going to get the wrong DNA. And lastly, it can, on the occasion, it will take up the gene of interest, what we're looking for. The jackpot has been hit. And so the question is how do we know what is what? How do we know which, when we're doing this, what has the plasmid of the gene of interest, what has the wrong gene, what has the original plasmid, what didn't even take up a plasmid at all? And what this results to is a screening problem. We're having difficulty figuring out what is where. And how do we solve this? Well, one of the ways that we can solve this is by making the right choice of plasmids or cloning vectors. And I think he just briefly talked about the term, that's not good. Stabilizing back there, briefly talked about the term vector. And vector is just a fancy word for a carrier, an epidemiological carrier. And so when we're talking about, like, say, malaria, the carrier of it is a mosquito. When we're talking about Lyme disease, the carrier of it is a tick. That's supposed to be a tick. And then we're talking about plague, bubonic plague, things like that. The carrier of it was a flea. Fleas were the first to get it. They bit the rats bit you, whatever, or the fleas went from rats to you, whatever, you die. You get the plague. Very brilliant drawings, I know. And so when we're screening those plasmids, what makes them right? What makes us these good choices? Well, we got to have one type of plasmids that are resistant to antibiotics. And all bacteria, like we talked about in the front, that they can exchange this stuff naturally. They can just, whenever they're bored whenever they're in the right environment, they can exchange plasmids. which is what I talked about when I drew this picture. They can exchange plasmids that can enable them to be resistant to types of antibiotics. And this is where you can get, within hospitals, usually a larger percentage of the bacterial population that are resistant to antibiotics. Because of this, they can introduce genetic variation in this way. And this actually, I think there was some game that I played that it was a computer game. Whenever you're sitting in class and in BCIS, it sucks. But the object of the game was to create a biological warfare virus that could wipe out the human race, and I would always choose a bacteria whenever I win, because bacteria can introduce genetic variation this way. And then one of the things that they can introduce in this is our plasmids, or that offer antibiotic resistance. I also want to point out, when we're talking about plasmids, being they're only 5% the size of a massive eukaryotic chromosome, and I say that term lightly. They're accessory genes. They're not essential. This has to have this to be able to be alive. It's nice to have, but it's just considered accessory. But anyways, I'm getting off track here. Our plasmids offer resistance. So like AMP sub R, superscript R is resistant to ampicillin. Tet subscript R, superscript R is resistant to tetracycline. Met is resistant to methicillin. Pen is resistant to penicillin. What we do, we're going to be able to use all these to help kind of, I guess, weed out the incorrect bacteria that got the wrong plasmids with a bacteria that got the right plasmids. And another thing that we can do is, all bacteria have something called a lac operon, and as we talked about in the last series, an operon is just a group of functionally related genes. So they're all a group of genes that code for some type of enzyme that helps play a role in breaking down lactose. Particularly, though, there's this gene called the LAC-Z gene. And now the LAC-Z gene is going to help. It's going to code for a protein that's going to become an enzyme. Enzyme in this case is a protein called beta-galactosidase. And what this is going to do is this is going to break the glycosidic bonds between glucose and galactose. And what another thing that is useful for this is that beta-galactosidase can be exposed to something called xgal. And xgal